Hello children, we have a question about the cell cycle here. Let us see that in cell cycle we have three phases G1, S and G2, that is in interphase. Interphase is said to be the resting phase, but it is the most active phase of the cell cycle. It occurs about 23 hours out of 24 of the cell cycle. It occurs 95% of the total interphase. This interphase has got three phases, subphases G1, S and G2. So the significance of S synthetic phase is that in S phase chromosome the DNA replication takes place. In S phase, that is in synthetic phase, DNA replication takes place. Remember, DNA replication means the amount of DNA gets doubled. That is, amount of DNA gets doubled. We have one example that before replication, if it is 2 picograms, before replication, if the amount of DNA before S phase, if the DNA amount is 2 picograms, now after S phase, that is after replication, the amount will be 4 picograms. That is, the amount got doubled during the S phase. But the surprising thing you should remember that the number of chromosomes will not get doubled. Number of chromosomes remain same. But the number of chromosomes remain same. That is the case here. So the number of chromosomes remain same in G1, S and G2. But inside the chromosomes, the amount of DNA gets doubled. For example, let's take here in any organism G1, S and G2. So let's take in an organism, let's take here the DNA and the number of chromosomes. So let us suppose in G1 the amount of DNA, let me write here amount of DNA is, let me take here 8 picograms. Then during S phase, amount of DNA will get doubled. It becomes here 16 picograms. During G2 also, amount of DNA remains same to that of S phase. So it becomes here 16 picograms. That is, amount of DNA is same in S phase and G2 phase. If you see the number of chromosomes, let's take here the number of chromosomes are 14 during the G1. Then S phase also they remain same 14. In G2 phase also they remain same, that is 14. So I have taken an example. It doesn't belong to any organism. Just an example I have taken here. The number of chromosomes remain same in G1, S and G2, whereas the amount of DNA gets doubled from G1 to S remains same from S to G2. So by seeing this, you will understand the question. A fruit fly is called as Drosophila melanogaster. It is a modern tool in the modern genetics. It has got 8 chromosomes, that is 2n number in each cell. During interphase mitosis, the number of chromosomes in G1 is 8. What would be the number of chromosomes after S phase? See, he is saying here 8 is the number of chromosomes in a G1 phase. After S phase, that is G2, the number of chromosomes remain same. He is not asking the amount of DNA. So within the chromosome, amount of DNA may get doubled, but number of chromosomes remain same. So answer will be again the same, that is 8 only, first option only. So this is something that they want to test your knowledge, what happens to amount of DNA during interphase and number of chromosomes during interphase. So that is the point of discussion here. That's what he want to test your knowledge of understanding of the interphase. So this is right, a very good question. He may give you, instead of fruit fly, he may give you other organism like pisum, where 2n equals to 14, allium 2n equals to 20, where maize 2n equals to allium 16, maize 20, human beings 46, that we know very well. So he may give the number of chromosomes varied according to the organism, and he may ask you the number of chromosomes in G1, S, G2, as well as they may ask you after karyokinesis, that is in telophase also, after cytokines also they may ask you. So this type of questions are nowadays being asked very much in competitive exams. One should have good knowledge of all the phases and what happens during these phases, then only he can do them very well in the objective competitive exam. So hope my explanation is betterment for you, helpful for you, and don't forget to subscribe the channel. Thank you.